The evidence for the deductive evidence that relies on homology to strongly support evolutionary theory is bolstered significantly in modern times by the molecular evidence that is now available to us. Since the sequences of DNA, the nucleotide sequences of DNA and protein, are really the documents of evolutionary history, we can compare DNA and proteins from various species, and based on the degree of similarity, we can infer homologous evolution from we can infer evolutionary homology. So, for example, human genes are much more similar to monkey genes than they are to, let's say, dog genes or bird genes or frog genes. And likewise, the frog genes are much more closely related to salamander genes than they are to any of the mammals, for example, or birds. And so the degree of similarity is reflected in homologous DNA and protein sequences in the what we'll call the documents of evolutionary history. And we can build phylogenetic trees based on molecular homology, just like we can build phylogenetic trees based on comparative anatomy homologies. So let's take a specific example. And what I'd like to talk about is a family of proteins called the homeodomain proteins. And these are encoded by genes called the homeodomain or homeobox genes. And what homeodomain proteins are transcription factors. And a part of their protein sequence forms a particular structure called a homeodomain structure, in which the protein folds into a characteristic shape containing 3 helices. So we'll change the color here. Containing 3 helices, which are just, we haven't gotten to this yet in the course, but we'll talk a lot about protein structure. But the point being that this homeodomain structure is conserved, highly conserved amongst a wide variety of members of a protein family. And these family members, these genes that encode these proteins, are found in a variety of animals, from worms to humans. And the homology of this family is quite strong, and we can compare different members of the family based on the degree of homology, on the degree of similarity, and deduce evolutionary relationships of the genes that encode homeodomain proteins. The homeodomain proteins have a helix on them, the 3rd helix of the 3-helix bundle here that constitutes the homeobox, the homeodomain. And that helix has amino acid side chains that insert into the major groove of DNA here. This is DNA. And they recognize particular nucleotide sequences of target genes, and thereby bind to those genes and regulate those genes, turn them on or turn them off in some cases. And this has led to some very interesting studies in which homeodomain proteins belonging to mice, a mammal, yeast, a single-celled fungus, Arabidopsis, a plant, and these different organisms that contain homeodomain proteins can have the genes that encode those proteins sequenced, and their sequence similarities can be compared. And interestingly enough, you find that there's a group of homeodomain proteins that are closely related based on their homology, based on their sequence similarities. And that group has members found in yeast, members found in plants, and members found in mammals. Where another group of homeodomain proteins are closely related to each other, again, found in those family members are found in mice, in plants, and in yeast. And what this suggests is that very early in eukaryotic evolution, an ancestral gene, an ancestral homeodomain gene existed. And that gene became duplicated. So there were 2 copies. And this copy gave rise, let's say, to this family. Let's call this the X family of homeodomain genes, whereas this duplicated ancient homeodomain gene gave rise to, let's call it the Y family over here. So here's Y and here's X. So this shows that there are genes in yeast that are more closely related to genes in plants or mice even than they are to other genes in yeast. And this is strong evidence for gene duplication being a source of variation that can be acted upon and can lead to the evolution of a variety of genes from duplicated ancestors. And this also points out the strong homology that has been conserved over long periods of evolutionary time between the ancestors that gave rise to yeast, single-celled organisms, and ones that gave rise to both plants and animals. So homology and sequence similarity can be used very effectively to deduce complex evolutionary relationships. And you will remember that we started out this lecture 1 with a slide that talked about the 3 domains of life, the eukaryotes, the archaea, and the bacteria. And the evolutionary relationships amongst these can be deduced very nicely by comparing the documents of evolutionary history, that is the molecular evidence that is found in their DNA, in the genes that they contain. And we'll talk a little bit more about the relationships amongst these domains when we get to when we talk about DNA later on in the class in a few weeks. Given that we've been talking about anatomical homology and molecular homology, here's an interesting paper in the journal Nature, the international journal Nature, a wonderful journal that if you get a chance to pick up sometime in the library, take a peek at it. It has lots of interesting articles in it in current biology and other fields of science as well. This recent paper in August of 2011 describes a new fossil that was discovered in China. And this fossil belongs to a shrew-like organism. We'll call it Jeremiah. That's the genus name. And this fossil shown here has been well characterized by these researchers. And it's very interesting because this reflects the ancient evolutionary history of mammals. For a long time, it's been known that there was an ancestral mammal that gave rise to 2 branches of mammals, the Metatherians and the Eutherians over here, the Eutherians. And these Eutherians eventually gave rise to the placental mammals, to which we belong, placental mammals. And the Metatherians gave rise eventually, it's a little more complicated than this, but they were the ancestors to, Metatherians were ancestors to the marsupials, like kangaroos. So the 2 major groups of existing mammals today, the marsupials and the placental mammals, were known to have diverged from a common ancestor. And DNA evidence suggested quite strongly that that ancestor lived, the divergence occurred, in other words, at about 160 million years ago. But the oldest Eutherian fossil prior to the publication of this paper, the discovery of Jeremiah, was dated to, based on paleontological fossil evidence, was dated to 125 million years ago. This fossil of Jeremiah then reconciles a previous discrepancy between the estimated age of a common ancestor and divergence of the lineages which would lead to marsupials and placental mammals based on the similarity or differences in their genes. That gave an estimate of an ancestor a common ancestor at about 160 million years ago. But there were no Eutherian fossils that were known that were older than 125 million years ago until this discovery. So now the discrepancy between the paleontological evidence and the DNA evidence is erased due to the discovery of this fossil. And paleontology only gets better through time. More and more interesting intermediates intermediate organisms are found that link together long lineages of evolutionary history. Now, what I'd like to do to finish up this first half of your first lecture is to talk about some of the unifying themes in biology by way of review and also by way of presaging what we'll be talking about as major topics in Biology 122.